All right, thank you.
So hi everyone, I'm Sivan, a MD PhD student in Morani School's lab, speaking to you today from Jerusalem. And my talk will be about human milk oligosaccharide utilization by infant gut Bacteroides species in general, and specifically about Bacteroides dorei. So first things first, what are HMOs? Human milk oligosaccharides. It turns out that breast milk contains a considerable amount of sugars called HMOs. All of them have a common lactose core that can then be elongated to create over 200 unique molecular structures. And the sugar structure can also be terminated with a fructose or sialic acid residue. But the real interesting thing about HMOs is that they're completely indigestible by infants, meaning that these sugars go down the GI tract of the infant completely unabsorbed, and they are then metabolized by members of the infant gut microbiome. So basically, breast milk allows the mother to control not only for the nutrition of the infant, but also for the composition of its microbiome community, which is pretty cool. Now, when it comes to studying HMO utilization by members of the infant gut microbiome, most research so far has focused on Bifidobacterium species, but we have good reasons to believe that other members of the infant gut microbiome are also able to utilize HMOs. Now, the immediate next candidates when looking for HMO utilizers are Bacteroides. First of all, they're well known for being good at complex carbohydrate breakdown, meaning that they can utilize many complex sugars. Also, we have some computational evidence showing an inverse correlation between the abundance of Bacteroides in the stool and the concentration of HMOs, hinting that maybe Bacteroides can break down HMOs. And also, in this important paper from 2011, we see an experimental evidence showing that the type strain Bacteroides tetaiomicron can grow on an HMO mixture almost to the same extent as Bifidobacterium infantis, which is considered to be the prototypic HMO utilizers. So for me, this figure alone is enough to convince me that it's important to study Bacteroides and HMOs. So if I want to talk to you about Bacteroides and HMOs or about Bacteroides doing anything at all, we need to first isolate those strains from infant stool samples. So ultimately, we would want to plate the stool sample on a selective plate to select only for the growth of Bacteroides. But what we found in the lab is that plating the sample on a non-selective plate, like a blood agar plate, before that helps with yields and with the recovery of many Bacteroides, of more Bacteroides strains from the stool sample. It makes sense because the samples are kept at minus 80 degrees, they're frozen, the cells are dormant, so it makes sense to get everything growing on a non-selective plates before making the growth conditions harsher and harder to grow on. Now, the bile estrogen plates, the BA plates, contain both gentamicin, an antibiotic which Bacteroides are inherently resistant to, and estrogen, a compound that they can hydrolyze to produce a black pigment. So black colonies on a BA plate probably indicate the growth of Bacteroides. And once we have the isolates, we can do whatever we want with them. We can grow them in liquid media, prepare stocks, sequence the 16S region to get a more exact identification, etc., etc. However, this isolation process is time-consuming and laborious. It takes time to plate. We have a lot of colonies on the blood agar plate that need to be transferred to the selective plates. So we would want to exclude irrelevant samples as soon as possible from this protocol. So we've found that we can add another step to this process of DNA extraction from a portion of the stool samples and Bacteroides-specific QPCR that allows us to screen samples prior to the entire wet lab workflow. So when we perform the QPCR screen on the stool samples, this is what we get. Please note that the y-axis is the minus log CT. It means that higher values indicate higher abundance of Bacteroides in the sample. And we can see a clear separation between samples that do not contain any Bacteroides in them here and samples that do contain Bacteroides. So this QPCR allows us to differentiate between samples that we would want to continue working with and samples that we do not need anymore. The positive control is a pure culture of Bacteroides fragilis, and the negative controls are cultures that are stool samples that were previously sequenced in lab and were found to contain no Bacteroides. And the last thing I want to say about this figure is that we compare the CD value with the Bacteroides specific primers to the CD value with general 16S primers, just to control for the general bacterial DNA content in each QPCR well. Later, we took the samples that passed our QPCR screen S4 to S10 and performed 16S RNA sequencing on them. So we can see that we indeed see Bacteroides in these samples. They contain Bacteroides. And as noted with the asterisks here, we managed to isolate various isolate strains from these samples. So our QPCR basically allows us to both screen for the relevant samples, and it's a good estimate of our ability to actually manage to isolate strains in lab. Okay, so once we have a library of Bacteroides isolates, we can characterize their growth on HMOs in our case, because that's what we look into. And when looking at the OD600 over time, we can see that some isolates do not grow on any of the test HMOs at all. Some isolates grow beautifully on all of them, and some isolates display a more interesting pattern of similar growth kinetics on similarly structured HMOs. For example, here the two pink lines represent sialylated sugars, 3SL and 6SL, and you can see that the growth pattern is very similar in both of them, which could indicate possibly a joint breakdown mechanism or pathway for similarly structured HMO structures. So it can get pretty hard to compare a lot of growth curves graphs for all the different isolates. So we can also use the AUC metric to quantify growth. AUC stands for area under the curve. And then we have a single specific value per strain per carbon source that can be summarized in a heat map. And again, we see that some isolates do not grow on any of the HMOs. Some isolates grow on some HMOs, and there's some variation across different strains. From now on, we wanted to dig deeper into the mechanisms that allow growth on HMOs, the genes, the pathways, the enzymes. So we focused on a Bacteroides dorei isolate that was able to utilize all of the test HMOs, and we performed RNA sequencing to have a transcriptional profile of its response to HMOs. Our analysis focused on GH genes, which stands for glycoside hydrolase. Different GH families break down different carbon bonds within sugar molecules. So each family is specific for a specific carbon bond. And when looking at B dorei's response to HMOs, we see an extensive GH family upregulation. Many GH families are upregulated. And another thing that we can see is that between similarly structured HMO pairs, for example, the fucosylated sugars, a lot of the GH families that are upregulated are shared between them. And same applies for the sialylated sugars and the neutral sugars. So when a lot of the GH families are shared, we can possibly assume that there is a joint or common pathway for the breakdown of similar HMOs. However, when looking at specific GH families, out of all of the upregulated GH families, here we look at the GH2, we see something different. We don't see a specific response to every type of HMO structure. Rather, we see that the genes are upregulated across all conditions quite consistently. So we don't see a specific upregulation of some genes for fucosylated or sialylated sugars or neutral sugars. It's more broad and consistent across all conditions upregulation. So the strain of B dorei that we've tested presents a generalized response to HMOs, which is surprising and different from what was previously described for Bifidobacterium species. Also, within each GH family, we see that some genes are upregulated in red and some of them are downregulated. And it could indicate a tailored response where some genes are actively expressed and some of them are repressed. Now, this applies for all of the GH families that were upregulated in the data. We have repeated the same analysis for various GH families. Here I'm only showing GH2 as an example. Okay, so to finish off, in the future, our next plan is to perform a knockout of gene clusters that were found to be important for HMO utilization following our RNA-seq analysis. We plan to do so with CRISPR, and we are now setting up the CRISPR system in Bacteroides in our lab. It's quite challenging, so if anyone has any ideas, suggestions about it, please talk to me. And that is it. Our lab always looks for new collaborations and new students, so feel free to check us out. And also, if you have any questions, you can contact me later or via email. Thank you.